Good morning.
So I think we can slowly start. I think there will still be some people joining, but anyhow, I'd like to welcome you all to this lunchtime workshop, Tools in Digital Pathology. There will not be a too large introduction. I have just one point that I'd like to suggest is that, so we have 5 minutes of presentation, 5 minutes of questioning and answering to our speakers. And I would suggest that we write our questions directly into the chat. So during questioning, I can efficiently read them out in the time that we have, and we can then hopefully get the answers a bit faster than if we try to switch between microphones. Yeah, with this, I'd like to welcome our first speaker, who will be talking about Paquo, or as it's also called, pathological Qupath obsession, a Python API for working with Qupath directly. And here to talk about it is the developer of it, Andreas Pohlmann. And with this, I would like to give over to you.
Thanks for the introduction.
So I think you should be able to share the screen.
Wait, I cannot yet. So let me work this out. I'm gonna just share the desktop here. And share, and let's start the presentation. Can you all see the presentation? Yeah, awesome. Okay, so my name is Andreas Pohlmann. Thank you for being able to present here today. I work in the machine learning research group at Bayer Pharmaceuticals in Berlin, and I started about a little bit more than a year ago now, and Paquo was basically one of the first things I worked on there, and I hope it's useful to you guys. So to give you a little bit of a background, what is Paquo? So Paquo is our, allows basically you as a Python developer to talk to Qupath, or in a way, intuitively manipulate Qupath projects. It integrates very well with your existing Python stack, so it's super easy to install, and you don't have to worry about basically the whole Java environment and everything. It also provides a whole bunch of quality of life improvements that makes working with Qupath projects, at least for someone who's familiar with Python, a lot easier. And with Paquo, you can read and write projects, you can read and write annotations, and also if you're whatever models you create, for example, create predictions in form of annotations and stuff, you can also write them back into Qupath projects. It's available on GitHub, and there's going to be another link in the presentation later. Okay, so a question might be, why did we build Paquo? So when I started, there was a project, a research project going on with pathologists that evaluated different annotation tools. And they picked Qupath because it had like the most featureful set of annotation tools. It's very performant, it's super well maintained, and it has a large user base. And also the user support is incredible on ImageSC by Pete Bankett. The hurdles we had was that basically our machine learning researchers and also the data scientists, they when they had to interact with Qupath, it was basically, Qupath is complex and it's written in Java, which is a hurdle for someone who's mostly familiar with Python. And it's then again also not easily interfaceable from other languages. So we decided to create a Python wrapper for Qupath. And for that, we used JPipe, which is a very cool library for for interacting with Java code from Python. So I recommend checking it out if you don't know it. So Paquo allows you, for example, to read projects. So here you see some example code where we, for example, read classes in the project. Super easy. We, from Paquo, import projects. We import a Qupath project and open whatever project in read mode here. And here you see we just access path classes, for example, that are stored in the project. If we want to access the images, they are also provided by a property on that Qupath project instance, and we can check if those locations are readable, because maybe some of the files you store are on a network share, etc., and they might not be accessible because it might not be mounted. And then you can also easily read annotations by just accessing an image hierarchy. That's the Qupath terminology, basically, that's here abstracted into the Python world. And you can then read out the annotations from Python in a very nice way. Paquo also allows you to write projects. And the cool thing about this is that Paquo only tells Qupath to write projects. So these are always going to be compatible with your Qupath. And here you see, for example, if we want to prepare a Qupath project with certain classes and maybe color coding so that the pathologists don't like make spelling mistakes or something accidentally in the class names. Here we define a few class names with maybe color, with the associated colors. And then we just use like a with statement here, like you would access a file object, maybe in Python. So it's very Pythonic in a way, the interface. And we just set the path classes to a list of these classes and add an image to our Qupath instance. And after that, we have a predefined Qupath project that you can just open in Qupath. And for annotations, it's very similar, but we have like a thing where there's shapely geometries, which is like the de facto 2D geometry thing for accessing geometries in Python. And it's very easy to add annotations to a project. So you might be able to mark a region that a pathologist should maybe annotate very thoroughly or something. And for our data scientists, because they are working with these projects, we added, because they all like working in Jupyter notebooks, so we added nice visualizations from within Jupyter notebooks. So you get good feedback there if you use it interactively. And we also added a simple command line interface, which here, basically, if you install Paquo, you get this command line interface that allows you to do some stuff very easily, like export all the annotations as GeoJSON, for example, from a project. And also it does stuff like here you see a video where we call that Paquo Quick View, where sometimes we had a workflow, you want to open an image and you have GeoJSON annotations stored somewhere on disk, and then it creates a temporary project that just has this one image in there and the annotations already provided so that you can easily browse it, which is just like a quality of life improvement. And if you want to get started, it's super easy. So as a user, you just install Qupath, you create a virtual environment for best practices sake here, and you just pip install Paquo. We have documentation at paquo.readthedocs.io that is very thorough. And if it's something missing, please let us know. We're always happy to improve there. And if you're a developer, just go to bioscienceforbetterlife.com/Paquo on GitHub. Feel free to start it. And if you want to contribute, we're super happy to get feedback and more and help. Yeah. And if something's not working for you, we're also happy to help out. And yeah, so I'm ready for questions if there's any.
So yeah, we have already one question in the chat from Andrew. He writes, "This is brilliant. I'm wondering." Oh, now my question shifted. I'm sorry. I'm wondering about speed efficiency. Are there certain use cases where it wouldn't be appropriate to use, for example, millions of cells?
Yeah, so I think we already basically the current implementation of the wrapper is doing things step by step. So there's like ways we can improve speed by going into other APIs from directly in the Java library, but we'd have to work that. I mean, I think we already tweaked it a little bit in that one GitHub issue where we could improve the handling speed by not updating the Qupath project every time. So yeah, we haven't used it for writing millions of detection objects or something in the project yet. But if there's any performance issues, open an issue and we'll look into it. Yeah.
We also have a second question from Rita Sarkis. Is the training or annotations only performed in Python and then imported into Qupath, or can these be performed in Qupath as well?
So currently, the only stuff we abstracted is basically reading and writing these annotations and detection objects back and forth between projects. So what we do right now is we create predictions outside of Qupath and then write them back into Qupath projects if we want to show them in Qupath projects to our pathologists. But we could, basically, it would be interesting to add features to Paquo to interface with the Qupath detection code so that we could actually execute these Java interfaces from the Python world. And so, yeah, it's like if someone's interested in contributing that feature, I would always happy to guide through and maybe sketch out a pull request to work on this. Yeah.
There's also a continuation of this question. Is the speed also limited by the size of the images? For example, if it's like TMAs, full slide scans, other heavy images?
So right now, for example, when you add an image to a Qupath project within Paquo, it takes the same amount of time it takes in Qupath to add an image. And for that, I think because, so it's either using OpenSlide or bioformats on the backend, sometimes loading these 4 GB images takes a little bit. So when you create a Qupath project with, I don't know, 100 images, it'll take a while. Like we already got rid of a bug that made it a quadratic and not linear. So now at least it has linear time to add the images, yeah.
Perfect. Are there any other questions? So then I'll ask one. So the what is kind of the extent of the wrapper's capabilities now? Are there any things that I could do with when I write Groovy script that I would not yet be able to do with Paquo?
So writing Groovy scripts within Qupath is always going to be more powerful than what is currently exposed in Paquo. A thing that is still on my, basically like in the pipeline that is like halfway done, but we haven't worked on it in a while, is we want to expose the scripting part also in Paquo itself so that you could load Groovy scripts also through Paquo, but it, basically, JPipe allows you to easily interface with everything that's in the Qupath Java world, basically, and you can then, like, adding new features would just be translating your Qupath your Groovy script then maybe to Paquo if it's needed in Python. Then, yeah. It's mostly a convenience thing ultimately for someone. Does someone need to learn Groovy now to do a certain thing in Python?
Good, wonderful. Thank you very much for this very interesting talk. I will definitely also try and integrate it in some of my pipeline. Thank you very much.
Thanks.
So next up, we will have a talk about Cytomine. The talk will be given by Raphaël Marais from the University of Liège. And yeah, with this, the screen is already there. Perfect.
Okay, good morning. Can you hear me?
Perfect, yeah.
Okay. So hello, I'm Raphaël Marais from the University of Liège. I initiated the Cytomine project in 2010. So today I will give you an overview of the main feature of Cytomine. It's not an easy task in 5 minutes, but I will do my best. So probably one of the big difference between Cytomine and other tools presented today is the fact that Cytomine is originally a web platform. So it allows you to collaborate over the web, over a regular web browser. And so you are able to share images, annotation, algorithm, and results over the web. And so basically the idea is that once you install your Cytomine on a server in your institute, you are then directly able to share all your data through the web with your collaborators. So you can give them access to images. You can allow them to collaboratively annotate these images. And then you are able to use these manual annotation to train machine learning or deep learning models that can be run on computing clusters. And then these algorithms can generate results that the user can proofread on the web user interface and then generate new results over the web. Of course, Cytomine can also be installed on a desktop or laptop, but then you will use the main collaborative feature of Cytomine. If you are interested to use Cytomine, you will not be alone because we have more than 20,000 users over the world that are using Cytomine either in research or in education settings. And there are, in addition to my research lab at the university, there are 2 other entities that can help you. There is a Cytomine cooperative that provide basic support and maintain the open source project and community. And there is also a corporation, which I'm not directly linked to provide services on top of the open source tool. And so we are, just to let you know that we are also involved in a 6-year project. So we are continuing the development of Cytomine through this project and other projects. With Cytomine, you can organize your imaging data set into projects and give access to a specific user to this project using the user authentication and access rights. We are supporting many digital pathology formats and other microscopy formats. You can visualize the wall slide images through the web browser, such as in OpenStreetMap and Google Maps. You can zoom in and zoom out using tiling system. In Cytomine, you can annotate these images, and we call annotation. These are regions of interest to which you can associate many different types of metadata, among which ontology terms that are vocabulary corresponding to the types of cell or tissues that you can encounter in your wall slide images. And you can use them, for example, to train, you can use them as classes to train your machine learning models. But you can also associate other types of metadata, such as key value properties, tax file, text descriptions, etc. And you can do this collaboratively, so each user has its own layer of annotation, and you can aggregate the annotation of the multiple users into the web platform, and you can send information over the web and emails, etc. One of the new features we developed this year is to be able to link image and annotation and using a side-by-side viewer, so you can synchronously visualize different images, such as different immunohistochemistry stains, and you can link annotation so that you can correlate the analysis of these annotation over different images. Another feature that we are developing, because often the digital pathology researchers are combining digital pathology slide to other modalities. Is the fact that we are releasing in a few weeks the new image management system that is very flexible, so that you can extend it easily to support new format, new slide scanner format, but also beyond the digital pathology world. And we have also support for spectral imaging data. Cytomine is highly extensible. We are using a web API, Python, Java client, and JSON standards, and also container technology to allows you to import and export all the data from and into Cytomine, but also to integrate algorithms from any other tool. So that's a development we made last year that allows you basically to plug workflows that are analyzing your data using major image analysis platforms such as ImageFG, IC, Cell Profiler, etc. And it's not limited to the list that you see here on the slide. It's very flexible. And you can, with this new software management system, you can then execute these algorithm through the web interface, but also visualize and compare the results of these algorithms on the web interface. So you can share the results with your colleagues around the web. And so here you see 4 side-by-side images. On the top left, you have the ground truth, and then you have 3 different algorithms that have been applied to the same image. We also ensure reproducibility by using this container technology and by saving parameters, values, version, source code, etc. And all this also allows you then to make benchmarking. And so we also offer benchmarking directly on the web platform with traditional metrics for benchmarking. Cytomine is documented. We made a major effort also this year to have a better documentation website, and especially for data scientists. So if you go to doc.cytomine.org, you will have access to this data scientist guide where we explain the main principle, and we provide a step-by-step example to integrate your own algorithm into Cytomine. If you want to know more, we have a Twitter account. Of course, the source code is on GitHub. It's Apache 2 license. And we have the website with the documentation. And I want also to let you know that this year we made 2 webinars. It's more than 1 hour long webinars. And the second one is specially dedicated for data scientists. So with this, I'm open to your questions. Thank you.
You're still on mute, Cedric.
Yeah, I just realized. Thank you very much for this interesting talk. We have a couple of questions. The first one is, which image reader is used to visualize the images?
So up to now, it was mostly OpenSlide, but with this new Python image management system that we have developed and that will be released in the coming weeks, it will be anything you want. So we are already using OpenSlide, but also bioformats and anything you want, because it's very flexible new system that allows you to define the reader for each of your formats.
Another question, is it possible to batch download a project, all annotations and images directly?
Yes, we have an API and we have the clients that allows you to download. You can filter by user, by project, by image, by annotation term, by anything. You can also batch download annotations generated by a given user or by a given algorithm. Everything is filterable, and you can do this.
Yeah. Is Cytomine used in the clinics or incorporated within routine reviews to share annotated images?
So our 2 main types of user are researcher and education, but we are starting to use it in clinics, but we are not yet there, I would say. But maybe if you have a more precise use case, I can answer to that.
So we have 2 more questions in the chat, but it is already time to switch to the next presentation. So if you want, you could also answer them maybe in the chat quickly, then we could move forward, if that's okay with you. And with that, thank you very much for giving us an introduction into Cytomine. And the next presentation will be about Ilastik, which will be presented by Rita Sarkis from the EPFL.
Thank you, Cedric. I'm sharing my screen. And okay, can you see it now? Okay, so thank you again, and I will be presenting Ilastik. I'm not the developer of Ilastik. It's a tool that was developed in 2011. It's been 10 years by Anna Kreshuk and her team, and now it's being held at EMBL. Ilastik is a tool for an interactive segmentation that doesn't need any knowledge in machine learning or an expertise in machine learning, but is heavily trained for users to supervise an annotation in order to perform different annotations. In my case, I use it in 2 contexts, for pixel classification and for object classification, but it has many other features that I'll be showing towards the end of my presentation. But I will highlight these 2, I think, strengths of Ilastik, the pixel classification and object classification. This is how it looks like when it comes to an interface, whereby we can see that we have different features of classifying our object or of interest or pixel of interest. That goes either from pixel classification to object classification or to tracking in 2D and 3D, carving, and even cell density counting. Talking about pixel classification and the input of images inside of Ilastik, ideally the HDF5 format is highly encouraged, but we can use TIFF or JPEG images. And then for pixel classification, we need to first select few features. And by default, as users, we are highly encouraged to select all the features that will help us to have a good classification of these pixels. As an output from the pixel classification, we will have like a probability map that we can extract and then threshold if you want, or use it in Fiji or another software for more quantitative analysis or further down analysis. Or we can use it as a background or basement to perform an object classification. I will elaborate on here the pixel classification by showing a quick tutorial, and I apologize for the quality of the video, but the main message can be seen. So first you need to point out to your folder where you want to have your project. And once you are inside of this, you need to select your images or your data set. We highly encourage to have a bit of variability within your images to be able to train them, because this is the training set of images, and this is how the model will be trained to detect your pixel of interest or different compartments that you want to annotate. But once you select your images, you can then after select the different features that are within Ilastik to be able to have a good segmentation. And as I mentioned before, by default, we highly encourage to select all these features. Once this is done, this is here the role of the user, because the user at this stage has a role to define or to label the images. And when it comes to the labels, it's up to the user to decide how many labels they want, depending, of course, on the images. But here, for example, in this image, we have we can clearly see the nuclei versus the background. And when we annotate, we can annotate different images or different annotations within one images, and then it depends on how the Ilastik will take it. We can then furthermore orient the annotation. It's highly recommended that the annotations are close to each other, so the software will know well how to decipher the limit between 2 objects or 2 pixels, or the limits between the compartments you want to target. But once we feel that, okay, we have a first run of annotation, it can be done on 2 or 3 images, and then we can move on to predict. And the prediction can be done in a live manner. So when we live update, we can directly see how Ilastik will classify our 2 labels, or will label based on our labeling that we performed as users. And the good thing with Ilastik, if we are not satisfied with what is being done, or if we see an incorrect labeling, we can correct on the spot and then update again, and then we see how the tool will learn with the user. And as an output from the pixel classification, as I mentioned before, we can have this probability map that we can then save to use it for an experimental set of images, or to use it for an object classification. And if I move on object classification, and here it will be also quick. So you can classify based on pixel classification if you feel that in this case, for example, you can decipher different differences within the nuclei. The first is to import what was annotated with the pixel classification. And then with object classification, we can modulate quite few parameters in terms of filters and size, or filters that will help us to define boundaries between two nuclei or cells, and then move on with trying to integrate what you want to have as metadata from this object classification that you want to include. And then to improve, so first you can improve the pixel classification, and second you can add on top of this classification an object classification. So we were able to get these objects, and then we have two different categories within the same object. So we can again label and then annotate and then predict in live and then correct and iterate. I would say the thanks of Ilastik is in this part of user friendliness and user being the one handling the annotation and prediction. And as I mentioned earlier, that it has many other features other than pixel and object classification, like carving, to be able to extract objects from 3D volume in a semi-automatic way, or counting. It can count densely packed cells. And with this, I highly encourage you to read the paper, the Nature Method paper from Ilastik, to visit their website. They usually organize tutorial or workshops very frequently. And even on their YouTube channel, they have a lot of tutorials for the different features that you can perform as user in Ilastik. And with this, I am happy to take questions.
Yes, thank you very much for your talk. So we already have 2 questions. So how noisy can the images still be, and the learning still be successful? The images you showed here are fairly clean in the sense that the background cells are nicely separated. What about with less specific biomarkers?
Yes, it can be really noisy, and when it's noisy, you need to annotate more. But when you annotate more, it's pretty much robust. I use it for human bone marrow H&E images, and it's not pretty much trivial in H&E to see the difference between different background. But when it is well trained, it is robust. For me, I know that Qupath also integrated this pixel and object classification, but I still rely on Ilastik for this feature. I feel it's more robust, or has a better background in this side.
So I worked a lot with detecting lymphocytes, and they can come in very densely packed regions. How is it when there's when you want to kind of separate cells that have a large overlap between them? So they can be like background cells and foreground cells. How well does it would work in such a case?
It works in a good way when you use the object classification, because here you can put as user your stringent parameters to be able to decipher. I didn't work with super densely packed cells that are different from it. Usually they are within the same class, but it's how stringent or how strict you are in your parameters that you define in the object classification. This can really help to orient the annotation how the user wants to see it. Yeah.
Good, and then we have one last question. How large can the images be that we can work with?
Yeah, it's a really good question. For the training, it's better not to have very large images. And if you have very large images, it's best to segment them. But for the experimental set, once you trained your model, you can use very large images. Like for me, I use 3 to 4 megabyte images. They highly encourage to use an HDF5 format, and they have within Ilastik a place where you can convert your image format to HDF5, because with this format it's easier to deal with big data with heavy images. Yeah.
Good, wonderful. Thank you very much. With this, we can go to our next talk. It's about Visopharm. For this, Simone De Brood from the Wet Swiss faculty here at the University of Bern will give us a quick introduction.
Thank you, Cedric. And hi, everyone. Yes, I'm based here in Bern, and I actually ended up using Visopharm software because I am a pathologist, a veterinary pathologist, and unfortunately with limited IT skills. So I looked for a tool that I can use to assess slides, ideally in an automated way, as well as having a deep learning classification available. And I have started using this software in spring this year. So I thought maybe it's best to show you an example of, I think you should see this, okay. Yes, what I'm trying to assess and where I think the software performs quite well. You may see that's the working space, and there is an option to have the slide, obviously, where you annotate or select any region you would like to assess. I find it
Simone, can I quickly interject? So we see the reference screen. Maybe you can make it
sorry.
quickly a bit bigger for us to see.
Yeah, sorry. No, that's not the right. No. Yeah, I think now, is it no?
Yeah, perfect.
Yeah, perfect. Sorry about this. So obviously you have the slide here to work with, and I find it rather useful that you have the option to work in studies, obviously using different folders, and this enables me to create the same images, for example, for different features, and to also distinguish between training sets and actual analytic sets. They are working with a kind of apps. So this is a protocol, an analysis protocol that obviously we can adapt to whatever we need to look for. So the first step, obviously, that I'm needing is a kind of tissue detection app that's fairly easy done. And you can see this is the way that these protocols appear. I can, of course, adapt any parameter here to the need, including the classification method. For simple things like that, it's sufficient to use, for example, decision forest. And of course, I can also see, depending on the feature, which actual channel, for example, color channel is best used to distinguish that. What I find rather useful is that even after processing, I can manually adapt and add some steps, how to improve the recognition and how to create the labels. This can be things, very simple things like, okay, change from an annotation into region of interest. The region of interest, of course, then at a later time point, again, can be assessed. I can exclude small or large structures, kind of perform an erosion of all the selections and so on. So you can see, and they're quite different steps. And that's just for the first thing to actually detect the tissue. So I would get something like this that's obviously working very quick, and I can of course do all that in batches, but you can already see there are things that can be improved, and I'm still trying to figure out how much is best done manually. So basically I would go in, check all the slides, do some minor corrections, or sometimes it's actually quicker to improve some of the performance by retraining, and then I would avoid that. I would then go, for example, to a next step where typically we are interested in only a certain part of the tissue to have it assessed here in the lung. And the idea was, for example, to exclude any larger blood vessels and any conducting airways. And for that, I could see that it's working best if I use the actual deep learning classification. I can, of course, see how well it learns, and it's very much, of course, depends how quick it learns and how good it performs, depending on what we are looking for. But that's an example here where it did a very good job already. So I would then exclude certain regions. Again, I decide what do I do with these regions. The reason why I tend to still keep some structures with the name and obviously distinguish them and not just declare them all as background is because I never know if at a later stage I actually do some analysis on exactly some of the structures that I initially excluded. So that's an option. And by the way, of course, just by ticking and unticking, I can decide each time which step I actually want the software to perform. So what are things that are happening here? Again, I try to turn all the regions that I want to exclude into regions of interest that would then, look like that. Obviously screening the entire lung. By the way, this is the field of view that you're seeing here. So going close, I would get a distinction between blood vessels and conducting airways. And then at a later time, I decide what I want to do with that. So already again, that's the time when I usually need to go in and check how well is the software performing. And that's the same story. Sometimes I can fairly quickly improve things like these by just rather than annotating much more, rather training longer. That's like what works better. And depending how frequently I have some mistakes, I may sometimes be quicker to just go in and manually correct that before I run the next app. Something that, for example, at this step may be done is before I continue with the measurement, I may already just have a program where I only have output variables. That's of course possible. So I would not classify anything. I would rather just get some variables that I later on can use to know how much of the tissue did I assess and so on. And then I would actually go to the analysis I am looking for. Again, you may see that I changed the method. Now I want to make sure that I can distinguish between certain features that can easily be distinguished based on the pixel values. So I would distinguish between a lumen space and the actual tissue. That's done again using this. And of course, I can quite individually decide what do I want. Do I want to classify certain values into scorings and make them visible? This will create kind of heat maps on the slides. That's quite nice to illustrate sometimes pattern of lesions. And of course, again, I can here turn on and turn off things that I want to include or exclude. And of course, then to actually have measured. That's only a selection. So in this case, we looked at the actual individual alveolar lumen of lungs. And we just tried with first measurements of like perimeter, diameter, area, and so on. But there are several more new variables, of course, that can be included. This would look like that. So this is taking then a bit more time, even though I'm fairly happy with the speed. And the positive thing is I can run again this all in the background as a batch, and I can continue working without problems. So I would get about this image and I would know the values for each individual area here that I am calculating. That's another point. Obviously, results are very easily to be exported, primarily as tab separated values, but also directly into Excel, especially with having many values. This can be essential. And that's about the summary after now the first few months that I'm seeing. So obviously I can confirm I can be an ignorant in the IT and I can perfectly use the software. The deep learning classification is definitely helpful for what we are doing. And as said before, batch processing is being able to be run in the background. And maybe one of the main reasons why I have to admit I changed from Qupath to Visopharm is because I have an excellent actual personal contact to improve the training. And that's maybe the drawback. So sometimes I realized, especially when we look into many small structures, primarily many small regions of interest, it may be slow and you need definitely a special hardware to actually make the deep learning run. that's from me. So I'm happy to take any questions. Thank you.
Yeah, thank you very much. So I think we still have some time for a couple of questions. So Andrew asks, this seems quite full-featured. Are there any things that you think are missing from the software or are frustrating for you to use?
Yeah, so something, of course, it's now a matter of what is my lack of still expertise using the software and what isn't. But we have already seen some things that I'm not very happy with. As mentioned, now I have these tons of data. As soon as I go for very small features, for me, that starts to be too slow. So a speed is a problem, but it's really for high, heavy data stuff. And for the actual measurements and annotations, I am really happy. Even though what you mentioned before with dense nuclei, there is a method how I can separate those, and that's really working quite well. And then it comes to the same, probably like always, the slide quality is first priority. And as soon as you have more artifacts than anything, this means I have to train much more in recognizing and excluding artifacts, and that's the same problem. So, but if the slide is of good quality, so far I have looked at a really wide variety of tissues and lesions, and I'm very happy. So I will continue. I know that. Thank you.
Maybe a quick other question. So can you export the classification or quantifications?
Yes, so that's a thing that I am a bit unexperienced, and I know they told me there's a way, but I have not even tried that. So I am aware that obviously this would be ideal to play around with what I'm doing or transfer it to other images, and I have not done that yet. So not sure how well they perform with that.
So there's one last question, but we are already a couple of minutes behind schedule. So I think we will go to the next presentation, but maybe you can also quickly answer that question in the chat.
Thank you.
So for our next presentation, we're going to hear something interesting about Qupath. And for this, we have Kaner Erkan from the University of Basel that will talk about Qupath.
Yeah. Hello everyone, thank you Cedric for introduction. I'm Caner, I'm working in University of Basel as PhD student in Pathology department. I'm not a developer of Qupath, I'm just a fan and also a user of a Qupath. And at the beginning of my talk, I would like to say we have already many softwares, many solutions for image analysis area, and for many things, for having many formats, many options always. The question is, why do we need one more software? Why do we need this Qupath, another software? I guess the most important reason of having Qupath, the use of Qupath is none of those softwares are designated for pathology, and then most importantly whole slide images, because whole slide images are very difficult data. There are very gigantic images. What we are seeing is very a kind of zipped data, but it's normally very huge, and then to read this data is not possible by all of other softwares, and using in pathology analysis. With this reason, using Qupath is a big importance. Besides, tissue microarrays are very important in our practice in research, and in Qupath, one can easily handle these TMA images and also analyze them, export the data, analyzing data from the TMA tissue, and then it's very convenient. Otherwise, also annotating those or evaluating those TMAs manually, including this data, again the data stored measurements in Qupath, and then afterwards getting a one file containing everything from there. And then another very important feature of Qupath is working in the project, because in the projects we always have a bunch of images, data, and then we need to work on them, and then we are making similar tasks, similar scripts we have, you need to run on them. And then in Qupath, one stores all the images from each project in together, and then Qupath just generates one file, contains the data related with these images, and then the original image stays somewhere else. And then in this project entry, this project data, we can, it's all the staining related information, all the objects, annotations, what we are making, all of them are stored over there, and then one can easily export afterwards related with each staining, each slide's related information so easily. And then this can be again combined with TMA information previously I told about. And then these objects and annotations, they are also in a have their own hierarchy inside, and then over there one can easily handle all those detailed tree of annotations very well, and then they can be stored and then analyzed separately. Separate annotations, separate tasks can be run for different type of the region of interest, for different type of structures which we generate, or they are detected by built-in functions of Qupaths. And these measurements, they can be generated in Qupaths, like the staining intensity related features or shape related features. They all can be stored in those specific objects we can you can see here, and then this information can be used afterwards different type of tasks, like classification of the objects. I will mention it a bit later. Besides using these objects, we can use our scripts, and then all those scripts again stored in that project files. And then classifiers, you can generate classifiers in using Qupath, and then they all are stored in those projects, and then they can be reused on other images, or they can be carried over other projects. And these built-in functions I mentioned before, these annotations, it's very important for making manual annotations on whole slide images. And then to be honest, my personal experience until this moment, Qupath always had been the most convenient software for me for making annotations. And whenever I needed to do any other tasks for any other software or platform, I could carry over those annotations over there, or I could export the data related from these annotation regions. And those annotations can be different times. It's very functional. It gives a good variety of the options for them. Besides this, the images can be processed inside Qupath. This can be fluorescent images or bright field images as well. And then these processed output of the output of this process can be used for the specific analysis separately. Cell detection, for instance, it's a very commonly used function of Qupath. You can see here, you can set up many settings over there. You can choose, for instance, your image, if it is generated by stain deconvolution inside Qupath, and then you can choose different type of the image you want to use for cell detection, and then you can get a quickly decent analysis for cell detection. And then afterwards, detecting those cells can be classified again with using another built-in function of Qupath. There are basic machine learning solutions available over there. One can use for segmentation, pixel classification, or object classification, as we can see here applied on those cells. And then here, while making those classifications, the measurements, the features of those cells, those objects which were again generated on Qupaths related to their shapes or staining intensity, can be used for making these classifications. And then this classification is quite interactive. If you are not satisfied, or you are spotting any errors, you can put more annotations, and then you can improve the AI model, and then you can see the live results updated here quite efficiently. Of course, if you are working very big data, this step can be a bit slow, but besides, it's going very well. Besides, you don't only need to work on the same slide for this. the beautiful part of working in the project. You can call the other annotations you made in the other images of within the same project by adding here inside the load training function, and then you can use all the information coming from all the project. The whole project can be used for training this data, and then you can increase your ground truth. And then for scripting, it's available for users from different experience level. One can simply just use the user graphic interface, and then Qupath tracks your movements over there, your actions over there, and then afterwards converts it into a script. It can be used quite efficiently. It's similar with macro recording function of ImageJ. And besides this, there are many available custom Groovy scripts available on the forums written by community or developers. You can see them in the forums in their website. I will share at the end, and then or you can using your IDE, and then you can write your own scripts. Another good feature of Qupath, it's not so much used, but it's very convenient if you are using, especially on computational clusters, command line interface. With this, thanks to this, one can use Qupath scripts in a headless mode, and then that script again run on the whole project or specific images, and then the data can be stored. I have to say this function can go a bit buggy. I couldn't check the performance during the last update, but this is maybe a bit more have space, bit more improvement in Qupath. For instance, this machine learning cannot be used, training cannot be used on command interface. This is, in my opinion, an important con of Qupath, but still it gives a big opportunity to user. For visualization, these measurements I mentioned before, these measurements can be seen by user here, all stored in each image, each object, and then afterwards this can be visualized by using the map, or it's already stored in a table on the Qupath, and then one can afterwards use this information or convert this information into histogram just to observe the changes, observe the features. And stainings, the channel viewer, is a very important feature for fluorescence images, multiplex images. One can easily select specific stainings to see the slide images, and also these channels can be generated, can be calculated by Qupath for bright field images. Sorry, the image is missing here. And then you can see from your hematoxylin, eosin, or immunohistochemistry slide, the calculated channel can be seen, and then afterwards those channels, calculated channels representing hematoxylin or DAPI channels from immunohistochemistry, or hematoxylin or eosin for hematoxylin eosin images, can be used for the further analysis on Qupaths. And another very important feature of Qupath is it gives a good opportunity, good platform to interact with other softwares. If you feel yourself more confident, or you want to use ImageJ for a certain problem, you can easily use this macros of ImageJ on Qupath, or within the script you can use some of the ImageJ functions within Qupath scripts. Stardate, it's a great tool, great machine learning based image object detector for cell nucleus detections. And then there are already pre-trained models, and then they can be used in Qupath. It's great. Or you can train your own Stardate model on Python and bring it to Qupath, and then use it over there for cell detection. Omero, it's a great platform for image repository. There is publicly available image repository, or your institute may have an image repository using Omero platform. And then since the last update, you can directly access these platforms, these servers on Omero or Python. As we at the beginning see, there is a good opportunity to use other platform, other language on Qupath, or make it to carry your information, your data, your images, or your just tiles of from your images to other platform and then analyze them over there, like carrying them to Python or to ImageJ or cell profile. It gives a very big opportunity to work on still whole slide images and making the analysis on other tools, softwares. This changing opportunity is really great. And then documentation of Qupath is really great. At the beginning, I'll show the links. The websites maintained by Pete Bankett, the developer of software, and his team can answer many of your questions regarding to image analyzer or Qupath related issues. And there are some short cons, of course. There are things it cannot do. You cannot manipulate your images, so your pixel values cannot be changed over there. This is a function you can use on image, you can do on image, but Qupath is focused on visualization and quantification. Basically, 3D images cannot be used. It's only focused works on 2D images. Ilastik time tracking, it's available. You can import those images in Qupath. Qupath, I didn't mention, but all whole slide image formats can be used in Qupath. It's very easy to work with them. Ilastik time tracking, also these functions can be available. This can be visualized, but it's not the best tool for using them. The other options, the other tools might be better if you have this type of analysis. Yes, thank you for listening. And then in this website, in these places, you can see the official documentation of Qupath, or you can see the questions of other users and again, answers from the community. Thank you.
So thank you very much for this thorough overview of Qupath and what it has to offer. So I think we have time for one quick question in the chat. If I can find it again here, yes. So for your workflow, are you keeping multiple Qupath versions installed to have matching versions for existing projects? Or are you updating projects when new versions of Qupath are released?
My personal usage, I can say in my computer, I have at least 3 versions of Qupath, and then I know which project I generated, which project in which of them. And whenever I'm changing between versions, I'm being a bit cautious. First, I'm making tests, I'm checking if everything is working well, and then afterwards, I'm making this migration. And for instance, for my main PhD project, I couldn't dare yet to update to new version. I don't know, I'm a bit conservative maybe because of this, but I didn't see mainly on forums so big issues between these images. If there is something, it's already documented by developers, and then they are saying which functions cannot be used. At the early stages, for instance, between 0.1 to 0.2, there was big changes, and then there was this issue, but I guess between 0.2 and 3, there wasn't so much. I didn't heard at least.
Okay, perfect. Thank you again very much for your presentation. And I think with this, we're actually coming to the last part of this workshop. And for this, Andrew will talk about the software suite that he is currently working on.
Thanks. So I think most of the folks in SDiPath are probably familiar with these tools, but I'll briefly run through them to conclude this meeting. So we built 3 tools. The first tool is for quality control. We basically realized that once you have thousands of slides being generated a day, it's not reasonable to try and manually go and review all of them to decide if the slide itself is an anomaly, if it's an outlier, or if there's kind of artifacts in there that should be avoided in computational analysis. And most importantly, we came to realize that quality control is not reproducible. So this is a pretty recent paper from ours this year, where we showed that even given the same whole slide images, and we asked 3 different people to go and pull out the good ones to be used for an experiment, we found that their overall concordance was actually quite low. But when we used HistoQC, that concordance went up sometimes in the case of over 20%. So this is a very interesting idea, because if you think about it, if you have irreproducible quality control, so different people start with different data, then naturally you'll have irreproducible experiments. So we really need to kind of have a consistent input in order to have a consistent output. And this is kind of the basis of all sciences in general. And we're hoping to bring this to digital pathology. So for that, we've developed HistoQC. It has a Python backend and an HTML5 front end. It computes various artifacts and then allows you to kind of interrogate your data set. It is free and publicly available at HistoQC.com. This is kind of what the user interface looks like, where we see each of the slides that have been analyzed at the top and the various metrics that have been computed. We could see here the parallel coordinate plot, where each blue line is a slide we've analyzed and each y-axis is a metric that we've computed. And you have a very nice ability here in these red circles to kind of start to see things like batch effects that might be present in your data. Or here we have one that has pen markings and the pen marking filter has identified it and said, hey, you might want to basically look at this. So this plot gives you the ability to have a very nice high-level summary of what particular things require additional quality control or additional review in your cohort. In the bottom, we can see the original image here on the left, and then next to it, a kind of fuchsia overlay created by HistoQC to indicate where it thinks there's good quality tissue for downstream computational analysis or human analysis. Although this is kind of focused on digital pathology, we have a imaging-based version as well that works on PET, MRI, CT. That's also freely available at this link here. Move on to the next tool, Quick Annotator. I think this is quite similar to the Visopharm presentation. I think the main difference here being that this is an open source tool. So maybe the support is not as good, but it's significantly cheaper. It basically begins by the user annotating some images. Here you can see the user goes and selects this image, then starts to annotate here in the bottom right-hand corner what they're interested in. At the same exact time, we're training a deep learning classifier to start to make predictions based off of the user annotations. You can see these predictions here now in this kind of white overlay. In general, it starts to do fairly good in the beginning. Now the user can immediately see where in the image the classifier is doing well and not spend time there. So basically not waste time annotating things that are not going to help the classifier improve, and instead sees immediately where the classifier is struggling. So that person can spend more time invested there. They can also import the annotations and quickly clean them up if needed. And then that basically feeds back into the model. The model is retraining, and then the annotations become better over time. We also see that we're able to provide increased specificity. So in this epithelium versus stroma use case, we can see that even though the user annotated regions like this on the top, Quick Annotator is able to go and provide a level of specificity that realistically wouldn't be feasible for a human to perform in a reasonable amount of time, right? So maybe this is 1 minute of effort. This is probably 10 hours of effort or an hour of effort, whatever it is. You can see here that the suggestions made by the tool are actually very accurate. So we don't have to make that investment, and we spend 1 or 2 seconds to have this level. This is also free and publicly available at quickannotator.com. There's a longer presentation available here. I think the main takeaway is that the speed up is significant, especially when you have many small objects. So in the case of annotating cells, you expect about 2 orders of magnitude speed improvement by using this kind of active learning, deep learning approach, while still retaining very high precision in terms of what you would be able to perform manually. And in some cases, not really evidenced by this F-score, you're actually able to perform better than what you would hope to do in a reasonable amount of time with your own hand. Lastly, we have a tool called Patch Sorter. The idea of Patch Sorter is basically once we've identified a bunch of objects, we would like to then subsequently label those objects. So maybe we've identified a bunch of cells, and now we want to go and identify where the lymphocytes are inside of that particular image. So Patch Sorter accepts as input the output from Quick Annotator. So you can annotate a bunch of objects. You then input those objects into Patch Sorter. Patch Sorter then goes and takes those objects and embeds them in a 2-dimensional space based off of their presentation, with the idea that similar cells are now plotted near each other. Hopefully here, by doing that process, the user can lasso certain areas and very quickly review large numbers of cells at the same time. So at least for me, none of these look like lymphocytes. So with a single button click, you can just say, all of these are not lymphocytes. Okay. And then that's going to further train the embedding space to try and improve the clustering. You can see an example of that here, where now if we look at this blue cluster, all of these look like lymphocytes. With a single click, you can say, yes, all of these are lymphocytes. So now instead of having to basically spend 1 second for 1 million cells to say, yes, lymphocyte, yes, lymphocyte, yes, lymphocyte, you can lasso areas. Maybe there's 5,000 of them at the same time. Say, quickly look, all of these look okay, yes, and now you've assigned a label at that kind of group level as opposed to an individual level. And we're seeing about at least a 60% improvement in terms of labeling efficiency by taking this approach. So I think that kind of brings my talk to a conclude here. I have the links I mentioned here. You can download that. Patch sorter is kind of ending the late alpha stage. And if you're interested in trying it out, I'm happy to have that conversation and perhaps give you the code so you can test it out. Thank you. I'm happy to take any questions.
Good. Thank you, Andrew. So we already have one question. How well does it perform when it comes to annotate the intercellular regions?
What is exactly, I guess this is for Quick Annotator.
Yes.
What is meant exactly by intercellular regions?
Like ECM or fibrous structures.
Yeah, so that's a great question. So the Quick Annotator is not some fantastic new technology, right? The back end of this still uses a UNet, and the benefit of using that UNet is that it's basically at this point been used, I think, for pretty much every single type of segmentation that we can imagine. So if you want, fibrotic regions, you can highlight fibrotic regions, it'll find fibrotic regions. So all of the papers that we've published recently in the kidney space, we've looked at tubules, peritubular capillaries, glomeruli, all of those use a UNet. And as a result, all of those would be perfectly acceptable to use within Quick Annotator to help kind of bootstrap that process. I will additionally add that the way that we designed this tool is that each of these backend components, so for example, that deep learning network, is very easy to replace by someone that's somewhat well-versed in Python. So we've designed it to essentially, if you can write a script, and we provide templates of these that will accept an image and produce an output, you can really replace it with anything. So maybe you don't like a UNet and instead would like to use a HookNet. If you already have code that uses a HookNet well, it shouldn't be more than a few minutes of effort to kind of retool it for these different algorithms and not have to change anything in the front end, which is more kind of JavaScript, and a lot of folks don't have experience with that.
Good. We also have a second question. What about the performance of quick annotators for not that black and white lesions? So, for example, indistinct borders between healthy and diseased tissue regions.
Yeah, so I think the question here is Quick Annotator supposed to go and do all of this for you, or is Quick Annotator supposed to make you more efficient at doing this? So if we think about a regular project, the user would have to sit down and basically annotate everything, right? So you have to go and find all those boundaries between these normal ones and these diseased ones and all this sort of stuff, and that's a lot of effort. I think the main purpose of Quick Annotator is to help you do that in a more efficient way. So in the beginning, it's not going to perform very well because it hasn't been subjected to many of those examples, but you will spend that time to go and correct that annotation. Now, is that lost time? Probably not, because you would have had to have annotated it anyway. So now instead of annotating it, we'll say directly in Qupath in a kind of a linear manual way. Here, you still have to annotate it if it's done poorly, but you stand the chance of that classifier being able to pick up on that signal and then incrementally get better. So over a period of time, the sky's really the limit. You can absolutely obtain regular UNet level performance, but maybe your data set is actually already fully annotated by the time you get to that point. So there's kind of a trade-off there, but regardless, I see no loss of time in doing an annotation in this manner versus trying to do it manually.
Good. We have one more question. Once the annotations are performed, these can be saved and then used on untrained images if these are similar images. Is that correct?
Yeah, so that's a great question. So we really designed this to be as user-friendly as possible. And if you look in the output directory, in the project directory, there's actually a folder called masks, which are already our PNG images that are binary masks. So you don't even have to do the export process. We intentionally saved all of the, we'll say, interesting outputs of the program in formats that you should already be comfortable with. So it's not like you even have to go and tinker around with things. It really is just copy those images and they're ready to go. We also provide code, for example, in Patch Sorter and Quick Annotator. They use smaller networks that are, I would say, in the order of maybe 100 to 400,000 parameters. And that's just so you can use it. For example, I can use it on my laptop. But as well, you can train it very quickly. So it's going to take a few minutes to train instead of, let's say, a week to train a 20 million parameter model. Now, I think the value there is you get feedback a lot faster, but we provide code that accepts all of the outputs from these tools. So the Quick Annotator outputs and the Patch Sorter outputs, and accepts those as inputs. And then you can go and say, now I'm interested in training a very robust model. I'm willing to wait 3 days for that training to take place, which is typical for a regular deep learning experiment. And you can click run on that and then end up with an even more robust model based off of the inputs that you provided.
Okay, wonderful. I think that was the last question so far I can see in the chat. I think with this, this concludes this workshop. I would like to take this opportunity, though, to thank again all of the speakers for their very interesting presentations today. And yeah, I think with this, also thank you very much for all who attended, for your attention today. And maybe if we get some positive feedback, we will also do this again sometime. Wonderful. Thank you very much.
Thanks, everyone.
And
take care.
Thank you, everyone.
Have a good day.
Everyone, have a nice day.
Thank you.